Hello, viewers. Welcome to Biodrunks. Let's learn some aspects of biotechnology. For your reference, I'll be providing notes, PDF link in the description. This video is also available in Hindi and Telugu. For more videos and continuous learning, please like, share, and subscribe. Let's start.
In the previous videos, we have done generation of DNA fragments for cloning. So where we learned how we can generate different DNA fragments, what are the different methods to generate them. In today's video, we are going to learn construction of cDNA library. So in the previous video, we also learned about how a cDNA is being created. In today's video, we are going to enhance that learning and learn more about it. So what is a cDNA? cDNA is a complementary DNA. Now, what is the cDNA library? cDNA library is a population of bacterial transformants or phage lysate in which each mRNA can be isolated from an organism and it is represented as cDNA insert. Okay, insert in any plasmid or a phage vector. Now, there are several steps for the cDNA library preparation. The basic steps which I'm telling you is suitable organism we need. First, we have to select a suitable organism. And from this organism, we are gonna isolate the mRNA. Once we get an mRNA, what are we gonna do? We are gonna use reverse transcriptase enzyme and we are gonna create a cDNA. This cDNA has to be inserted into the vector and then to the host cell where it multiplies and creates multiple copies of cDNA inserts. So as according to my definition, it's a population of bacterial transformants, okay, in which the cDNA insert is present in a plasmid or a phage vector. So as you've seen, these are the basic steps. Let's get clear with each and every one step. So the first step which we have is isolation of mRNA. So you are going to select a suitable host or a tissue. Okay, that's the first and foremost thing. It should have excess of mRNA. Now, we have different procedures for that. First and foremost, the basic procedure is chromatography. So what does this procedure do? In chromatography, we use polyuciferose. So when it has a polyuciferose, what does it do is poly A tail. If you all remembered, mRNA has a poly A tail. So this poly A tail, it binds with the poly U of the chromatography and hence the mRNA is being retained. You can also use density gradient centrifugation to get huge amount of mRNA. Okay, so here are the basic methods for isolation of mRNA. Now, what is the next step? The next step is we are going to prepare the cDNA. So what is cDNA? It's a copy, or I can say it as a copy or complementary DNA. It's a copy or a complementary DNA, which is produced by using mRNA as the template. So here, what is the template? Here the template is mRNA. Now, okay, see now, this reaction has been preceded by an enzyme called reverse transcriptase. So it's the most vital enzyme in the cDNA process. Okay, so what is this reverse transcriptase? It is an RNA dependent DNA polymerase. Okay, and this particular enzyme was discovered by Temin and Baltimore in 1970. So it is obtained from a microorganism called as avian myeloblastosis virus. Okay, so this is the whole process. Okay, so how the about reverse transcriptase now. Now let's talk about the process of mRNA to cDNA and then cDNA library. Okay, see, already you know most of the topics, like how it has been done. This is an image which has been taken from the author, that is Panel Mathis Hairbus. Okay, I found this to be very much related and it could help me to easily explain you the concept. So now, as you know, this is an mRNA. Okay, so this is your mRNA. This mRNA has a poly A tail. Okay, now we already know what is the primer we need here, right? So as it has a poly A tail, now we easily can get a primer which has a poly T tail. Yes, so we got a primer. This is the primer which is having a poly T tail. Now, as it has a poly T tail, what is the next step? The next step is the DNA is gonna start, okay, replication, or I should say polymerization. So here annealing is starting and then you have the next level of cDNA. This is it. Okay. Now we have it, and always remember we also methylate it. Okay, I'll tell you why. Now we get a RNA and cDNA hybrid. So this is a RNA and cDNA hybrid. So reverse transcriptase helped us to synthesize the cDNA here. Now I don't want this RNA here because I want a DNA, pure DNA. So what I'm doing, I'm using RNase H. What does it do? It digests the mRNA. So now this RNase H, it digests the mRNA and you are left with a single-stranded cDNA. This cDNA, it does not need any primer and it serves as its own primer, and once it gets the free OH, it synthesizes the another strand. So as you can see, it's forming a hair loop structure here and it's synthesizing. This hair loop structure can also be broken down by S1 nuclease. Okay, so this is the one which you are having. This is the cDNA. Okay, now this cDNA is methylated. Why is it methylated? See, methylation process, it protects against the internal cleavage. Okay, so you're not going to have any internal cleavage when you're using, because you might be using a same restriction enzyme and there might be some sites that might be present in this. So we are methylating it so that it doesn't get cut. Now, this your double-stranded cDNA is being inserted into a particular vector and then transformed into a host cell. So in this video, I'm not talking more about vector and host cell, as in the upcoming videos, I'll be describing it in more detail. Now, till here we have done. Now let's see what's the problems we face. Okay, so the main problem is this mRNA. If you're not getting a proper mRNA, okay, so this mRNA might lead to incomplete copying. So if you have this incomplete copying, that means your half of the DNA is wrong, missing, half of the information is missing. Okay, now if you are using a nuclease here, like S1 nuclease, to break the hairpin loop. So what happens is, during that, then it might nibble away some ends of the duplex. Okay, so this is another problem. Always remember, whenever you are doing incomplete copying is the main problem which has been observed. Okay, now another thing is not only the incomplete copying of mRNA, there is an incomplete copying of cDNA, that is a single strand, into a double-stranded. Okay, so these are the most basic problems that are being observed while your cDNA is being prepared. Okay, now let's learn about the next thing. What is the next thing we are going to learn? Is the properties of cDNAs and cDNAs libraries, and why are we using it? What are the applications of cDNA library? See, properties are very easy. It's the basic. As eukaryotic cDNAs, they are free from intron sequences. Okay, so intron sequences. Because mRNA is consists of only the functional group. Now, another thing is it has smaller gene. Now, when you compare a cDNA sequence with a genome sequence, it permits the delineation of introns or exon boundaries. Okay, and always remember cDNA is enriched with a lot of mRNAs. Okay, it is enriched with lots and lots of mRNAs, but it may contain only a few or no clones. Okay, that represents rare mRNAs. So it might also have rare mRNAs with it. When you compare it with a genome library, it's very different. Okay, so in genome library, you have only you have all the sequences, but when you see cDNA library, only structural genes are being transcribed here. Now, what's the next applications? So where are you going to use the cDNA libraries? See, cDNA libraries is very important because we have only the expression sequence present in it. Okay. Now, it can be used in different ways. The first and foremost thing is, we know it as it has expression sequences. We call it as expressed sequence tags. Now, what is this expressed sequence tags? See, because it has most of the expression sequences and it's been replicated, or I should say, we are getting cDNA from it. So it is the functional part. So we can use this for different processes, such as we can use it for physical markers. We can use it for identification of genomic clones. We can use it for gene expression analysis. Okay, and we can use it to design the PCR primers. Okay, there are so many. Okay, there are many of them. These are the basics. So hope cDNA was clear for you all, and the construction of cDNA library.
This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to Biodrunk. Let's meet in the next video. Bye-bye.